Okay, let's discuss the citric acid cycle, or sometimes it is CAC, or the tricarboxylic acid, or TCA, okay, or simply as known as the Krebs cycle. So we recall that the Krebs cycle, or the citric acid cycle, or the tricarboxylic acid cycle is the third stage in the biochemical energy production. So this is common for all types of metabolism. Either it would be protein metabolism, carbohydrate, or lipid metabolism. So this is the third stage. So we discuss first the common stages before we elaborate the individual metabolisms. Okay. So the citric acid cycle, this is a series of biochemical reaction in which the acetyl portion of the acetyl-CoA, so ito yung acetyl portion niya, the two carbon atoms na naka-attach kay acetyl-CoA is oxidized to a carbon dioxide. So makakaproduce tayo ng carbon dioxide and the reduced coenzymes FADH or FADH2 or and NADH. Okay, so ang mga produce dun sa citric acid cycle are carbon dioxide, the NADs and the FAD, or the NADH or the reduced form, and the FAT H2 or the FADH2. Okay, so always remember, in order for it to be considered as the reduced form, may mga hydrogens siya. Okay, mas maraming hydrogen. And of course, this cycle is discovered by Hans Adolf Krebs. That's why it's known as the Krebs cycle. Okay, so let's have here the first reaction, which is known as the condensation reaction. Okay, so for the condensation reaction, so since this is a cycle, the product of the last uh, reaction from the previous cycle will be used. So ito yung product dun sa eighth reaction, which is the last reaction in the previous, yung naunang cycle. Gagamitin siya to react with the acetyl-CoA that will enter the cycle. So you need acetyl-CoA to enter so that oxaloacetate will be formed into a citrate. Okay? So, isa-isahin natin tong condensation reaction. Condensation reaction, the first reaction, needs an oxaloacetate which contains 4 carbon atom, and oxaloacetate is considered to be a ketodiacid kasi nga it's a ketone. So, ito yung ketone group niya. And diacid because it has carboxylic group there. And of course, kailangan din ng water. So, papasok yan dun during the condensation reaction. Then, the acetyl-CoA, 2 carbon atoms lang ang reactive dito kasi alam natin na napakahaba ng reaction ng structure ng coenzyme A, but the active portion is just acetyl group. And ulitin-ulit natin that, let's recall that the acetyl group here came actually from stage 2, which is the acetyl-CoA formation, na na-breakdown from stage 1 kasi mahahaba yung mga carbon atoms ng mga carbohydrates, lipids, and proteins. Okay, so once na naging 2 carbon atoms na lang siya attached to the coenzyme A, so magkakaroon sila ng interaction kaya maproproduce na yung citrate, which is a 6 carbon atom molecule. So from oxaloacetate na 4, okay, ang carbon atom plus the acetyl-CoA na 2 carbon atoms. So, yung citrate, 6 carbon atoms na. So, tingnan natin that this is the acetyl group and this is the oxaloacetate. So, ito oxaloacetate, and the portion that was added here dito sa top naman yung acetyl group. Okay? So, with this reaction, meron tayong, aside from the production of citrate, which is a hydroxy triacid, so meron siyang hydroxyl group, meron siyang tatlong carboxyl group there. That's why it's a hydroxy, one hydroxy, and a triacid. Okay? So, matatanggal na si coenzyme A. So, kasi si acetyl lang naman ang na-attach kay oxaloacetate. And of course, the hydrogen and the oxygen will supply, coming from the water, will be supplied dito naman sa mga um, hydrogen part ng hydroxyl at saka nung uh, carboxylic part ng citrate. Okay, so magtatanggal din ng isang hydrogen ion during the process. And hindi lang dyan nagtatapos ang condensation reaction or the first reaction ng Krebs cycle. Meron din tayong dapat i-take down note na enzyme. For this condensation reaction, wherein the oxaloacetate reacts with water and the acetyl from the acetyl-CoA, kailangang magkaroon or there should be the presence of citrate synthase. This is the enzyme that enables this reaction to be possible. Okay, so that's the first reaction for Krebs cycle. Okay, so this acetyl CoA or so the acetyl group will be attached in this portion of the oxaloacetate so that ito ay form. Okay, citrate. Okay, let's now have the second reaction. Okay, so the second reaction will be isomerization. So from citrate, which is the product of the first reaction, so citrate will now become isocitrate because of the isomerization reaction. But this is not just a simple process kasi the citrate will lose water molecule para maging aconitate. Aconitate. Sorry. Aconitate. Okay. Kaya nga ang ating enzyme dito ay aconitase. So that the citrate will then become aconitate. And from aconitate, that would be the second reaction. So mag-a-add tayo ng water molecule so that aconitate will then become the isocitrate. Nevertheless, the citrate and the isocitrate are isomers. They are both hydroxy triacid. So, at parehas sila na, okay, palitin natin. So, if you're going to see, it still has the hydroxy there and we have the triacids. Okay. Pinagkaiba lang ni citrate tsaka ni isocitrate is that the hydroxyl group has been attached on this middle carbon atom. Samantalang itong si hydroxyl dito, nandito naman siya sa baba. Okay? So only water molecules has been removed and was again added during the isomerization reaction. So again, the enzyme used here is the aconitase. So pagbibilangin natin yung carbon atom natin for the second reaction, 6 pa rin, na-retain pa rin natin yung 6 carbon atom molecules ng mga intermediates. Okay? Now, for the third reaction, which is the oxidation and decarboxylation. So, during oxidation, laging tatandaan, gagamit tayo ng mga redox intermediates, which is the NAD or the NAD. Tapos, basta lahat ng oxidation reaction na makikita ninyo, merong mga NAD or FAD or NAD or FAD. Okay, so katulad nitong 6th and 4th carbon atom. Pag decarboxylation naman ng sinasabi sa Krebs cycle, there would be the production of carbon dioxide. So pag nag-produce ka ng carbon dioxide or paano malalaman na nag-produce ng carbon dioxide? So ito, going out yung carbon. So carbon dioxide. So decarboxylation, ibig sabihin in order for you to produce the carbon dioxide, kailangan magtanggal ka ng carboxyl group dito sa ating reactant. Okay? So let's have this third reaction, oxidation and decarboxylation. The enzyme needed here is the isocitrate dehydrogenase. So laging tatandaan that in Krebs cycle, when we use the enzyme dehydrogenase, usually that is for the oxidation reaction. And if that is dehydrogenase, of course, sabi ko nga kanina, we are going to use the redox intermediates, the NAD or the FAD. Okay, so we need here the NAD plus in order for that to have the NADH. So that's the reduced form. Okay, so what happens here in the structure is, so tingnan natin kung anong nangyari. From being a uh, hydroxy triacid, okay, naging keto diacid na siya. Yan, keto diacid. So, natanggalan ng carbon atom. Saan yung kinuhang carbon atom? Okay. So, here, this carbon atom was not there anymore. So, yan yung source ng ating, yan yung tatanggalin as na magiging carbon dioxide. Okay? So, whatever that was used, so makikita natin doon sa first reaction, gumamit tayo ng NAD to be reduced to NADH plus H. So, that plus H will then be reused again para matanggal naman si carbon dioxide. So, from isocitrate, which is 6 carbon atom, a hydroxy triacid, it will then become carbon 5 na lang siya kasi nga nagtanggal tayo ng carbon dioxide during the decarboxylation reaction. So, the oxidation reaction is the first reaction wherein the NAD has been reduced to NADH, and then the second reaction is the decarboxylation wherein the H plus will initiate the removal of the carboxylate or the carbon dioxide from the isocitrate. Okay? So, the fourth reaction, it would still be oxidation and decarboxylation reaction. So, progressive, no? So, from C5, mag magtatanggal na naman tayo ng another carbon dioxide. So, magiging C4 na lang siya. So, from a keto diacid, ulitin natin paano naging keto diacid. This is the carboxyl group or the ketone functional group. And then we have the diacid, which is the carboxylate. And then magiging thiodiacid na siya. Okay? Kasi nga, for this fourth reaction, we are going to use again the coenzyme A, and then that will be incorporated para maging thio. Kasi yung sulfhydryl na yan, siya yung mai-add dito. Yan. Ito na si coenzyme A, na-incorporate siya kay succinyl-CoA. So, from alpha-ketoglutarate, which is 5 carbon atom, after the second oxidation and decarboxylation reaction, or the fourth reaction of the Krebs cycle, with the presence of alpha-ketoglutarate dehydrogenase complex, magiging succinyl-CoA na siya. So, coenzyme A has been incorporated in this intermediate from carbon number 5, magiging carbon number 4, which is a thiodiacid. Paano naging thiodiacid ito? Again, we have the thio, which is represented by the sulfur, and diacid kasi may dalawang carboxylate group. So during the process, again, we are going to use the NAD redox intermediate para maging for it to become the reduced form NADH, which again, matatanggal na naman yung carbon dioxide. So pagtingin natin dito, kung kanina yung middle portion dito yung carbon dioxide na tatanggal during the third reaction, so this case naman, so ito naman carbon dioxide na to ang matatanggal. Okay? So, if you could still remember, yung nasa top nito, ito yung acetyl group. Okay? Ito yung acetyl group na na-attach kanina dun sa first reaction. But during the process, take note that hindi yung acetyl group na nadagdag during the first reaction ang nare-remove as carbon dioxide. Rather, yung sa oxaloacetate kanina, yung mga diacids niya ang natatanggal as carbon dioxide. Okay? So, this carbon dioxide will be this carboxyl group will be then removed as carbon dioxide. Okay? So, that would be the fourth reaction in the Krebs cycle. Now moving for the fifth reaction, which is the phosphorylation. So during phosphorylation, so from the root word itself may phosphate group dyan. So may magdadagdag tayo ng source of phosphate which is the GDP. GDP, hindi siya ATP ha. This is GDP. Okay? So with an inorganic phosphate, itong PI na to, that will then become the GTP. Okay? So from succinyl-CoA, which is 4 carbon atoms, and it's classified as a thiodiacid, okay? Ang mangyayari is the coenzyme A here will then be removed dahil sa tulong din ng phosphorylation, and the enzyme that will be used here is the succinyl-CoA synthetase. Okay, so the CoA from the succinyl-CoA will be removed so that it will then become a succinate, which is a saturated diacid. Okay, pag sinabing saturated, of course, lahat yan ay single bond and a diacid because of the carboxyl group there. Saturated because of the carbon to carbon single bond, not carbon to oxygen double bond. Kasi dito, etong COO na yan are drawn as a carbon double bond O and an oxygen. Pero pag sinabi natin saturated, of course, the carbon to carbon are single. Okay? So, the fifth reaction of the Krebs cycle actually produces a GTP, which is a high energy. Parang parehas lang din sila ni adenine, ni ATP, adenosine triphosphate. Pero dito si guanosine triphosphate, parehas lang sila, magkaiba lang sila ng nitrogenous base. Okay? So, that is the fifth reaction of the Krebs cycle. The sixth, kasi 8 nga itong reaction, no? So, for the sixth reaction, which is an oxidation, ulitin natin, pag sinabi nating oxidation, meron na naman yung mga redox intermediates. For this time it is the FAD. FAD will be oxidized to produce the reduced form FADH2. Okay, so from this reaction, kailangan natin ng enzymes in the form of succinate dehydrogenase. Okay, so from C4, succinate, ganun pa rin, walang decarboxylation dito. So fumarate will be formed, C4 pa rin siya. But from being a saturated diacid, because of the oxidation, magiging unsaturated diacid na siya. So, ito na yung unsaturated part, the carbon-to-carbon double bond, but still diacid pa rin siya, so meron siyang dalawang carboxyl group there. Okay? Then, the seventh reaction is dehydration. So, we need the use of water here, so water will be incorporated, plus of course, the presence of the enzyme fumarase, wherein the fumarate, okay, will then become a malate. From being an unsaturated diacid, magiging hydroxy diacid na siya. So, the unsaturated part will be broken so as to accommodate a hydroxyl group. Okay? So, meron siyang isang hydroxyl doon, tapos meron pa rin siyang dalawang diacid part. Okay? So, fumarate will then turn into malate during the seventh reaction of the Krebs cycle. And then the last reaction is the oxidation. Again, so oxidation na naman yan. Meron tayong dehydrogenase, but this time it is a malate dehydrogenase because the malate, C4, a hydroxy diacid will then turn into an oxaloacetate, which is a ketodiacid. So we need the use of NAD plus in order to produce the reduced form NADH plus H. Okay? So from the hydroxyl here, magiging a keto na siya. Magiging keto group siya. Nevertheless, diacid pa rin siya. Okay, so that oxaloacetate that would be formed during this 8 oxidation will then again reacts with any oxaloacetate, any acetyl-CoA that will enter again the succeeding cycle. So, cycle lang siya. So, kung ano yung oxaloacetate na na-produce dun sa 8 reaction, of course, makiki-interact siya dun sa acetyl-CoA na papasok during the first or the first or initial state of the succeeding cycle. Okay? So, let's just have this recap. meron tayong 8 reactions during the Krebs cycle. We have condensation, isomerization, and then we have oxidation reactions, dalawang oxidation and decarboxylation, okay? And then followed by the fifth phosphorylation, and then another oxidation, and then hydration, and then the last is oxidation. So, all in all, parang mayroon tayong 4 na oxidation reaction, may dalawang decarboxylation. Isang condensation, isomerization, hydration, of course, yung isa naman ay phosphorylation. Okay? So, pag titignan din natin, may mga water tayong ginamit, may water na ginamit, tapos nagtanggal ng water, tapos nagdagdag na naman, tapos nagdagdag na naman dito. Okay, so maka-cancel din yan mamaya. Meron din tayong mga na-produce na dalawang carbon dioxide. Ilang beses tayong gumamit ng NAD. Okay, so we have here the NAD, and then another NAD, and then another NAD. So, 3 beses tayong gumamit ng NAD. So, therefore, nagka-produce din tayo ng reduced form na NADH. Naka-produce din tayo ng GTP, which is an high energy molecule. And then, of course, naka-produce din tayo ng FADH. Isa lang. Okay? So, from here, let's have the summary of that citric acid cycle. Okay, so the chemical reaction of the citric acid cycle takes place in the mitochondrial matrix where the needed enzymes are found. So, nandun yung mga 6 na enzymes. Okay? Except for the succinate dehydrogenase reaction that involves in the FAD. Okay? So, kung 8 yung ating reactions kanina, 8 din yung klase ng enzymes na ginamit natin. So, let's recall that enzyme ends with ASE. So, we have here citrate synthase, the aconitase for the isomerization, and then we have the dehydrogenase enzymes, pero magkakaiba yung mga dehydrogenase natin. So, for the third reaction, we have the isocitrate, kasi nga isocitrate yung ating reactant here, and then we have the alpha ketoglutarate dehydrogenase, kasi ito naman yung gagamitin during the fourth reaction, and then the succinate, kasi ito rin naman yung ginamit para ma-fuel yung oxidation, and of course, the last is the malate dehydrogenase, kasi ganun din siya. Siya yung mag-fuel dun sa 8th reaction. So, meron din tayong fumarase, and of course, we have here the succinyl-CoA synthetase. Okay, so for every 8 for that 8 reactions in the Krebs cycle, meron din siyang corresponding 8 enzymes. Now, for the overall reactions, kanina sabi natin, pumasok yung isang molecule ng acetyl-CoA, gumamit siya ng 3 NAD, okay? NAD+, and then na-reduce form yan sa 3 NADH, okay? So, that's the 3 oxidation reaction. And then, the 4th oxidation reaction uses FAD. And then, na-reduce siya sa FADH. Okay? Then, during the phosphorylation, we have the GDP, and with the use of inorganic phosphate, kaya nakaproduce tayo ng GTP. Of course, we use 2 molecules of water. Na-cancel out na yung isa. Kasi, okay, kahit na 3 beses tayo gumamit, pero gumamit ulit kasi tayo dun sa, nag-remove tayo ng water during the isomerization, kaya 2 beses na lang. Okay? So, ito pala ay parehas sila dyan. Okay? And then we have here the production of the 2 carbon dioxide molecules kasi nga during the decarboxylation reaction. And of course, the CoA, okay, so ma-remove din siya as CoA. Okay? So let's have the important features. The fuel for the Krebs cycle is of course the acetyl-CoA, obtained from the breakdown of either carbohydrates, fats, or lipids. So yung acetyl part nila. So there are four cycles that involves the redox reaction or the oxidation reduction. And the oxidizing agent is the NAD plus, which is used 3 times, and then the FAD, which is used once. Okay. And then in the redox, the NAD is the oxidizing agent, okay, wherein the carbon oxygen double bond is formed. And then the FAD is the oxidizing agent wherein the carbon to carbon double bond is formed. Okay, kaya nga dito sa ating, okay, balikan natin dito, nagkaroon ng unsaturated ito. Ito yung sinasabi nilang pag gumamit daw FAD, merong carbon to double bond formed. Okay? And then we have, so from there, the three NADH and the one FADH2 will carry electrons and the proton ions, or electrons and hydrogen plus ions, that will actually be needed during the fourth stage, which is the electron transport chain, wherein masisynthesize naman yung ATP. So yung whatever NADH or the FADH2 that was formed during the citric acid cycle, sila yung magpupush dun sa fourth stage, which is the electron transport chain, which are actually the common reactions for the biochemical energy production. Now, number 5, the two carbon atoms that enter the cycle, which is a part of the acetyl unit of the acetyl-CoA, and the two carbon atoms that leaves as a two molecules of carbon dioxide, okay? So, pero hindi sila yon, are not the same. They are not the same. The acetyl-CoA, so ulitin natin yung number 5 na yan. Kung makikita natin, ito yung acetyl group from the acetyl-CoA, nandito siya sa taas. Ito naman yung part na ito na nasa baba. Pero pag titignan natin, okay, ang natatanggal na carbon dioxide is dito sa may acetyl, dun sa oxaloacetate, hindi yung acetyl-CoA group na yon. Okay, so nare-retain siya. Okay, so the two carbon atom that enters are not the ones that leaves as a carbon dioxide. Okay, and there are four, let's take note that there are 4 B vitamins that are used to function for the proper functioning of the cycle. So we have the riboflavin, which is in the FAD and in the alpha ketoglutarate. So that's the first B vitamin. The nicotinamide, of course, we discussed this, that it is included in the NAD. And the pantothenic acid, of course, that is a part of the coenzyme A. And now we also have the thiamine, which could be found in the alpha ketoglutarate dehydrogenase complex. So apat yan. Apat na B vitamin: riboflavin, nicotinamide, pantothenic, and thiamine. And of course, the last is we produce the high energy GTP molecule during the phosphorylation process of the citric acid. Okay? So, the regulation of the citric acid cycle, the rate at which the citric acid cycle operates, is controlled by the body's needs for the energy. So, the ATP. So, saka lang nagkakaroon ng Krebs cycle kapag kailangan ng mag-produce ng ATP ang ating body. Pero kung taas pa rin ang supply ng ATP, of course, that high concentration or high supply of ATP will inhibit, hindi niya i-stop niya yung cycle. Kasi nga, ang purpose ng Krebs cycle is for the production of the NAD and the FADH2 para magpunta dun sa fourth cycle, which is the electron transport chain, na kung saan dun naman talaga gumagawa ng ATP. So kung mataas pa yung ATP, there's no need for Krebs cycle to push through. Okay? So, okay? So, ganun yung ating mechanism for the control of citric acid cycle.